Okay, welcome. Good afternoon. And those of you in the Zoom audience aren't seeing me because our camera operator has COVID. So we're going to just have the screen share, but I hope you're seeing the screen share. And if not, we'll fix that. This is our first time back on campus. Glad to see the student faces in the room. And also always welcome to the extended audience, extended community on Zoom. So welcome all. We are back in person and we're all fully masked, and I'm glad to see you. It's way more fun here. So this does make me happy, even if the camera doesn't work to record Brian. So our guest today is Brian Bingham. Brian, I've worked with for years. He's one of my favorite faculty. He's both college and environment. He spends most of his time down at Shannon Point Marine Center, which if you don't know about, you should. If that map that you have of the Salish Sea had cool things on it, one of them would be the Shannon Point Marine Center. But it doesn't have points, it's just got a map. But now he's done such a good job down there that he's now the director of the Coastal and Marine Science Program. Max?
Marine and
Coastal Science Program, Max, MACS. That's right. So he's moved into admin and is putting his talents in mind, keeping that program a relatively new program. Again, if you haven't heard about the Max program, that's a cool thing to check out as well. Perhaps we'll hear a little bit about that. And then after that, so I'm just going to turn it over. And again, my apologies to the Zoom audience that you won't get the hand gestures, but at least you get the audio and the slides. Welcome, Brian.
Thank you. Well, thank you, Stefan. It's terrific to be here with you today to share some thoughts. Stefan asked that I take some time to talk to you about things that hopefully you'll find interesting, but also that have relevance to the Salish Sea and the environments that surround us. You just look off campus here and you're looking at part of the Salish Sea. So I'm going to share some stories, and they come from habitats all over the world in the different areas and places. But there's an underlying theme that when we're finished, I hope you'll see that these principles are critical to the Salish Sea and how it functions and to the oceans in general. So I'm going to start with this. This is a story about an invertebrate, and some would consider this an invertebrate villain. This is the crown of thorns starfish, Acanthaster planki. It's a starfish that occurs on the Great Barrier Reef, down Australia, up through Japan area. And it's a sea star. Primary feature here, these thorns all over the surface. And actually it's venomous. If you pick it up and those penetrate your skin, you get a nasty sting out of it, can cause swelling and some pain. They can get about 80 centimeters across, would be a big one, average size, a big dinner plate. They're considered villains because they eat corals, and we know corals are incredibly valued and they're in trouble. And one of these sea stars on an average day will eat, it puts its stomach out of its mouth, puts it down on the coral, digests its coral tissue, and it'll consume about the area of a picnic napkin, something about that size in a single day. Now that in itself is not terrible, but what is terrible, maybe,
No, we'll get there.
is that the slide won't change. Okay, thank you, Stefan. So what is terrible is that they go through outbreaks. And there's these cycles where suddenly scores of these things will appear on the reefs, and they'll mow through this reef and they'll destroy enormous parts of the reef. And of course, that's a big problem, especially if you rely on the reef for the products, or you're a scuba diving company, or you're some kind of resort that relies on it. So the local governments have put a lot of effort into trying to control the crown of thorns sea star. And initially, that involved people divers going out with knives and cutting starfish, and that doesn't work. They regenerate very well. So next they would take out gunny sacks and they'd load them in gunny sacks and drag them up on the beach and bury them. And they killed a lot that way, but that's labor intensive and they sting and they're heavy. So then they turned to chemistry. And initially that involved going out with formalin and injecting these things with formalin. Obviously that's not a great idea. Formalin kills everything. And so best practices now, the way they're using them currently, is to go out with vinegar. And that's a diver with a big bottle of vinegar and a syringe that he can just inject these things and it kills them. They fall off. And they've put lots of effort. Many crown of thorns starfish have been killed and removed from the reef. And the net effect of that has been it really hasn't mattered. Yeah, you can clear certain areas of the reef, but these things come back. And the next outbreak is just as big. And you see these corals that they've digested off in the background here, the bleached look. So in essence, it's really been a failure to try and control this species. It goes through its cycles, and it's going to continue to go through its cycles. And the question is, from a scientific perspective, why is that? Something as obvious and important as this sea star, why can we not control this animal? And I think the answer is that this is Acanthastra planki, but so is this. Many invertebrates, and in marine systems in particular, have what are called complex life cycles. And what that means is the baby doesn't look like the adult. They have a distinct larval phase. And that larva, a number of these would fit on the head of a pin, and they're up in the plankton, and they're floating around for some period of time. You know, aside from sharing the exact same genetic material, those are really two different animals, and yet it's the same thing that are intimately connected. And that's the case for many, many species. And here's a picture of the full life cycle of this animal. And like many invertebrates, it starts with a male and a female that spawn their eggs and sperm up in the water. They fertilize. They may go through 2 or 3 larval phases, and then eventually they settle down to the bottom. They grow up. They get to the next level. That's a typical complex life cycle. I want you to think about this a second and think, when one of these sea stars spawns and it throws these eggs up into the water column, how many of them do you think make it all the way around this cycle to reproduce? I want to do a little experiment with those of you here in the room for the kinesthetic tactile learners. And what I'm going to do is ask you to answer the question, what percentage of these things do you think make it to reproduce again? So I'm going to ask you to raise your hands, everybody. And when I hit a number that you think is a threshold, drop your hand. And nothing, no scores here. So everybody put your hands up. Okay, how many think 80% of these make it all the way around? 60%. Okay, some arms down, 30%. Okay, a few arms left, 10%. Okay, still 4 or 5 arms, 1%. Okay, we have 2 or 3 arms. Okay. Let's do a little math here. Let's say we dismiss class. I told Stefan, I need your people, and we all go out into Bellingham Bay, and somehow we do an exhaustive survey of the entirety of Bellingham Bay for one species of sea star, and we find 2 of them. In the entire bay, there's just 2 of them. One's a male, one's a female. In a single spawning season, one of these sea stars can actually produce 1 million eggs. Okay, so if we assume 1% of those make it around, that means when we go out next time to check sea stars, there's 1 million sea stars in Bellingham Bay. Second generation, we split those 50-50. And what's happening here? In that next generation with 1% survival, what is that, 500,000 million billion, 500 billion sea stars? I'll go one more generation just because I like saying the word quadrillion. You don't get to say that very often. But that's what it looks like after 4 generations, if you had only 1% survival. So 5th generation, this is planet Earth. Well, that doesn't happen. And so that tells us something is happening in this life cycle. And in fact, if we go back to that initial drawing of the life cycle, if we went into Bellingham Bay and that population stayed consistent, that means all that female has to do is replace herself and the male who fertilized her. So that means almost everything is not making it around this cycle on average. Only 2 of those eggs on average are making it around. If you doubled that, you've doubled the population. If you have it, your population has dropped in half. Now that's all crude averages and numbers, but it's a concept that is critically important to thinking about how these populations function and how they're managed. So the question is, where are the bottlenecks? Think about that complexity of that life cycle. There must be places where that pinches down, and where are they? Because if we can identify where they are, That gives us a leg up on conservation, on restoration, on control of pests, on a number of things that we worry about in ecology and restoration and conservation. So let's start at the beginning. Do they fertilize? As an ecologist, we used to always just think, yeah, of course they do. They throw them up to get fertilized. Maybe a few of them don't, but the question is, do they really fertilize? And there's some really, really creative work that was done with the green sea urchin, which is one of our local sea urchins. This is done out of San Juan Island, Friday Harbor Labs. And green sea urchins are really nice to work with because they're easy. And if you take one of these and you bring it into the lab and you give it a little shot of potassium chloride, it's not lethal. Give them a little shot of that and they spawn, which is really convenient because you can get sperm and eggs from these animals. And those are three females in the lab being spawned. And this researcher, Tim Pennington, went out and he spawned some sea urchins, and then he got some plastic petri dishes. And this was really clever. He cut off the bottom of the top and put Nitex in there so water would flow through. And then he loaded them with those eggs. And then he had a weight on the bottom and a fishing float on the top. And these were his fake female sea urchins. And he lined them up in the direction that the seawater was flowing. And then he went around, he got another green sea urchin, he gave it a shot. And if it was a male, he set it right at the starting point here. And the male starts to release the sperm. And then as that continues and as the male is releasing that sperm, it drifts down with the current, it dilutes, and it crosses over, presumably, these pseudo females. And then he could just collect those up and see how many fertilized. Really pretty simple idea, but pretty clever as well. So I want to show you what his data looked like. I think you might be surprised by it. So what we've got here is a graph that on the bottom shows the distance downstream from the spawning male, with the male right at the 0 point. And then the y-axis is what percentage of those eggs actually fertilized in those little petri dishes. And right at the 0 point, with that petri dish right next to the male, you can see there's 100% fertilization. Now, Pennington also put one female a meter upstream, and he did that just so if there was any extraneous sperm in the water, it would be recorded by that one. There's 0 fertilization a meter upstream. So there wasn't a bunch of sperm already in the water. It was coming from the one he had spawned. Now, what do you think the rest of that graph would look like? Now, if you're just forced to draw a line in there, what do you think that looks like? Okay, I see some people kind of a downward slope. That's what it looked like. It was stunning because it shows that within 25 centimeters, you're down around 20%. That's this far. That's that far directly in line with the spawning male. That's not even off the axis at all. By half a meter, you're almost done fertilizing. What that suggests is if these animals are spawning, they've got to be really, really close to one another. If they're not, you're not seeing fertilization of a lot of these eggs. Now think about how we manage things in marine systems. We do it, well, you can collect this many of this and this many of that. Would it be possible to reduce densities to a point that they're just physically separated far enough they're not going to fertilize? It's one of the reasons we have protected areas. We set apart areas and say, don't collect from here because we can keep the densities high. But the way we manage doesn't often use that. It's just, well, you can take this many, and there's potential at least that if these animals are already stressed on this, there's a bottleneck there in fertilization that could have some pretty important consequences. Now, that's sort of a best case scenario too, because what we know, and this is a picture of a fertilized egg with sperm all around the outside. What we know from experiments we've done in the lab is that the sperm of at least this species only last a couple of hours. Once they're released, they start to swim. They're diluted and they start to swim really energetically and they burn out pretty fast. And if they burn out, they're not going to be able to fertilize that female. And then we also know the number has to be high because only about 1% of the surface can actually accept a sperm. And so there's only a few points on that egg where the sperm can actually fertilize. So if your counts aren't high enough, the odds of actually getting a good spot are not high. And so that spatial and the temporal are really important. And we can go a step further and think about time also in how do these animals know when to spawn. If there's a 2-hour window, if you miss it by an hour, it doesn't matter. You've wasted everything. All the year of preparing your gametes has been wasted. But how do you know when to start preparing your gametes? There's some kind of cycle that tells, they spawn once a year. How do you know when to start that? And then when do you know when to conclude it? And it turns out for our shallow water urchins, it's probably light, the duration of the light cycle, and temperature may have some influence. But what about climate change and changes in our environments and the things that are happening that are changing the world these animals live in when they're so finely attuned to these processes? Could that be important? Another thing we know is that the sperm of these animals, at least, and many others, are very sensitive to pH. Sperm tend to be weak, and if you mess with that pH, they're not going to fertilize. And we know our oceans are acidifying, and it's that acidity that tends to be a problem. So again, if this really is a critical bottleneck for these species, as some of the data suggests, we should be concerned about these processes. Now, this is not an invertebrate, but it's another story that actually, I think, shows that it's not just these animals we should be concerned about, but fish. And this is a bluehead wrasse. And this was work that was done down in the Caribbean. And these bluehead wrasse are wonderful fish, and they're territorial. Males find a coral head, or usually a coral head, and they'll defend it and they'll protect it, and they live there. And then females come, and the females will visit the coral head. They spawn. The male sees the spawn. He swims up. He spawns on top of it, and his sperm only lasts seconds. After a couple seconds, few seconds, it's not going to fertilize. So he's got to be cued to when those females come. And there's a researcher who thought, I wonder if these fish are constrained by selective pressures to really focus that reproduction, like we've talked about with these invertebrates. Are there pressures on this process? And so what he did was he got a bunch of graduate students and he suited them in dive gear, gave them garbage bags. Being a graduate student is a lot of fun. You're out in the Caribbean with a garbage bag. You jump off the ship and you watch these fish, and as soon as they spawn, you run up and you grab the spawn. You carry it up to the deck, and then you filter it, and you count the sperm and you count the eggs. And the question is, are they matched? Because they saw that different sized females had different sizes of spawn. So does the male have any clue what's going on? The answer is yes, they do. They found a very, very tight correlation that these fish are very visual. And if they saw a larger spawn, they'd produce more sperm. Smaller spawn, less sperm. And so it again suggests there's some really tight selective pressures probably operating on these animals in the reproductive mode to make sure that fertilization happens. Well, that wasn't the end of the experiments. These folks are really creative. And one of the things they knew was that not all bluehead wrasse are created equal. And you have the really good alpha male fish that get the best habitats. They're the primo corals. They're the spots that are just nice. And that's where the ladyfish like to go. Then you have the weaker males out in these marginal habitats where maybe a fish will go, maybe she won't. But the good habitats may get multiple fish per day, and the marginal habitats get one, maybe two. You're a scientist, obvious thing, let's mess with that. And so they went and they captured the male off the good habitat, took him away and put a poor male onto that habitat and said, I wonder what's going to happen. And so sure enough, the next day comes and this poor fish is in a nice habitat, happy with his new life. And here comes the ladyfish and he darts up there and 100% fertilization. He's good to go. Then, whoa, here's another one. Ah. And you can see the fertilization drops to about 10%. And he's doing his best, but you can see that by the time that sixth fish comes, he's gone. And so he was unable to match that because he wasn't acclimated for that kind of a reproductive process. So then the question is, what happens on the second day? And they wanted to repeat this and say, is this a consistent pattern? And this is crazy. This is the next day. So this fish had totally retooled its reproductive physiology and its behavior. So he was ready. So the next day when all those fish came, he's maxing 80, 90% in all the fertilizations. So again, it suggests really fine tuning of these processes because they're so incredibly important for these animals. And so in this new habitat, he's now functioning as makes sense for him reproductively. Now, this comes at a cost, I should add. And that cost is he couldn't sustain it. He was dead in about a week. According to the graduate students, he had a big smile on his little blue head, but he couldn't do it. So that's a short-term experiment. The fact is that there's that reproductive cueing. So another question we could ask is, okay, we've talked about coral reefs and some of the habitats there. What about other habitats? Maybe really extreme habitats. And this is work that was done from deep water submersibles. And if you think about shallow water animals and the challenges they have, now think about deep sea. There is no light cue, there's no temperature cue, there's no salinity cue. How do they do anything? Not only that, but you're motoring around in a submersible and you may see an animal here and then here's one over there and then 5 minutes later you find one over here. They're not in these densities usually. And I found a couple examples here where we got pictures of animals who were clustered, but often they're not like that. So how do they do it? And what are they doing to try and cope with this? Do they have different processes, different needs? And this is one of the animals that was not uncommon there, a great little sea urchin. And they have these really long spines and they stand up on the sediment and they kind of walk like a big spider. They're really cool. And when they spawn, there's a single puff of sperm that comes out and kind of floats away. Now, sea urchins and many invertebrates have sperm that's highly conserved. Sperm cells kind of look like sperm cells for the most part. There are exceptions, but in general, they tend to be pretty conserved evolutionarily. They have a head that has the DNA in it. They have a lytic, an acrosome on the front end that dissolves through the egg and allows the DNA to be injected. And then they've got a tail that they swim with. That's what was expected of these sperm, of these sea urchins that have sperm that look like that. That's what was found. In fact, they did have sperm that looked like that. They also had these other funky sperm that were mixed in there in a high proportion. You go, what are these animals doing? What's going on here? Clearly, that's not going to fertilize anything. It's like trying to thread a straw with a spaghetti noodle. It just doesn't have the mechanical capacity to fertilize an egg. Now, we don't know, but I think the hypothesis is that these sperm here, called apirine, means they don't fertilize. Their whole intent is just to wrap up the eupirine, the fertilizing sperm. So when they secrete this or they release this, these are all wound up in a big bundle. And what that does is it prevents dilution. And so as this sperm cloud's moving along, it's preventing the fertilizing sperm from starting to swim and burning off their energy. And the hypothesis is that cloud then stays consistently at a fairly high density, high concentration, gets caught in maybe the spines of another one right above the gonopore where the eggs come out, and maybe that's how they're doing it. We don't know, but the truth is there's something going on here. And that's one of the really fascinating things about this work is every animal's got a story. I think every animal has some kind of story. We're just figuring some of them out, trying to learn how they function under these pressures. Okay, so that is probably a bottleneck for some species. That initial stage of getting those eggs fertilized. Has anything overcome that? I mean, is it a bottleneck for every species? And we don't know. There's a lot we haven't studied. What I've got here in the inset figure is that data from Tim Pennington, where he used the green sea urchins, showing that 25, 50 centimeters away, they're not fertilizing very much. And then the bigger one shows another animal that someone went out and did a similar study with. And you look at that, and we're at 100 meters and this thing's fertilizing. That is a quantum shift in capacity, which says this animal probably is not constrained by this fertilization bottleneck. What animal might that be? We don't know. It's that one. So could it be that the reason we have crown of thorns outbreaks is because they have surmounted this bottleneck? Could that be the case with other outbreak species? And it turns out these males, when they spawn, are just enormous bags of sperm, and they just produce enormous amounts of it, so that you just get these clouds that move down, unlike some of these other species. So interesting idea, hard to test, but an interesting concept. So if we argue that, okay, there might be a bottleneck in that first step of fertilization, then let's talk about the next step. Let's say they do get fertilized and they turn a larva out. And I've got 4 of our local species here, and the insets show the larvae of each one. All very different in behavior, in habitat, in feeding, in everything that they're doing, and probably subject to very different, well, undoubtedly subject to very different selective pressures in how they're working out there. So what about those? Is that a bottleneck? If you make it to the larval stage, is it a done deal? Are we good now that we're going to complete that cycle, or is it another potential bottleneck? And does it differ from species to species? And you might ask, well, do they eat? What do they eat? And do they starve? Is that a problem for animals out there? And in a changing environment, is that going to become more of a pressure? Are we going to see changes in that? And one argument might be, well, let's produce larvae that don't eat. That'd be a pretty good trick. And in fact, some are like that. This lower one is a tadpole larva of a sea squirt. If you go down to the docks in town, you look over the edge, you'll see these sea squirts that are plastered on the side, and they produce tadpole larvae, they're called. And those are lipid droplets in the head. The parent just loads it with lipids, and so it doesn't ever have to eat. It just swims around as long as it's going to, it lands, and it never has to eat anything, which has its advantages, but it must also have disadvantages because 90% of our shallow water species actually feed. So it's sort of the exception as opposed to the rule. And the argument is that if you don't feed and you have a shorter lifespan, you can't go as far, which makes it harder for you to spread your risk to other habitats. And so having larvae that feed may be a better strategy. Interestingly, deep sea larvae, many of them actually have to feed, which is even a bigger problem because there's no light down there, so there's no phytoplankton. So what are they eating and how are they doing that and surviving? So some of them probably starve. Well, what about predators? Is that open ocean a safe place to be? And that's a photo of a bunch of herring I took down in Belize, and enormous schools of herring moving through there. And you've got to imagine that not a lot escapes our attention when they're out there. And there's reports of some of these schools 1/3 of a mile long that moving along and just sweeping anything they can get out of the water. So certainly there are cases or locations where that could be a problem. A paper in 1973, I love this term, describes a coral reef as a wall of mouths. That's what it is. And as the water comes across with the tides, it's stripping out 60% of the plankton that's in that water. And so if you're a larva, hopefully trying to get back to where you can land and settle and take up life, you're facing this enormous suite of predators. It could be pretty dangerous. In our local waters, some of the things to think about: mussel beds. Mussels move a lot of water, as do oysters, as do most bivalves. They really can move water. It's been estimated they could be removing 100,000 larvae per day from the water. You have these big smacks of jellyfish. A group of jellyfish is called a smack. If you learn nothing else, that's kind of fun. They're out there feeding on things. We have barnacles out there. When you see those barnacles going like this is their legs actually out there sweeping the water and grabbing things to pull it into the mouth and eat it. All kinds of things. It's a dangerous place if you're a larva. You're small, you may have lots of resources that your predators want. So probably there is a fair amount of predation that goes on. But if you're a scientist, how do you document that? How do you measure that? And for years, people did it by putting larvae into bottles and culturing them. And then you throw a predator in there and you say, wow, they eat larvae, and they'll eat a lot of them. And there are a number of papers that talk about how many larvae were eaten and what predators did it. That's been pretty soundly criticized. That's a really clever way to do it. It said doing that's like measuring predation on Christians in the Colosseum of Rome. You put them in there, the lions are going to eat them. But it's so unnatural, we really don't know. And it's a tough question. do we know how much this happens and how important it is? Well, there's a really clever researcher by the name of Randy Olson who decided he was going to tackle this. And he was down in Australia, and he's working with an animal called a sea squirt, which up in the upper left-hand corner, that's the animal. And what Randy did was put on his scuba diving gear. He went down to the bottom, he laid on the bottom, and he laid there and watched one of those till a larva came out. And that's a picture of the larva at the top. It's called a tadpole larva. And then he followed it. Pretty crazy, but pretty interesting too. And he followed it and saw where it went, saw what it did, saw what happened to it. And his question was, do they get eaten? What are the chances these get eaten? And these are small, you know, he had to work at this. And these are some of his data. He did this at different depths down through the water column. And if you look at the top, that one about 4 meters says he followed, what is that, about 13 or 14 larvae, and 100% of them got eaten. Down a little deeper, looks like 1 or 2 of them survived. Bottom line is there's a lot of red on that graph. A lot of these animals were eaten, and very few of them actually made it past this suite of fish predators and coral predators that caught them. Now, that in itself is interesting, but the stunning part of this is the average time was 53 seconds. So within 53 seconds, these things are just blazed. They're just being hammered by the predators. And so very, very few of them probably make it to a point where they're actually settling. So for this species, that could be another really important bottleneck.
When do they qualify as survival?
When they settle, when they hit the bottom, these hit the bottom and then they attach permanently. So that's when he would consider them safe.
Do they not be predated off the rock?
Oh yeah, that's another question. And that's a great question because that's the juvenile part of that cycle. We're still in the larval phase, but you're right on. Really good point. Another question, how do they behave and where do they go? And this is down by the ferry terminal in Anacortes, the marine labs right there in the trees. Down here in this bay is a beautiful eelgrass bed, and it's premium habitat for Dungeness crabs. And not only Dungeness crabs, but reproductive Dungeness crabs. So the inset shows a female with an egg sponge on her abdomen. And they're very common in this habitat and may be really, really important to the numbers of Dungeness crabs that are out there. But the question is, if you got a bunch of Dungeness crabs in there and they produce those larvae could be in the water for 3 to 5 months. So this population is not reseeding itself. So where do these crabs come from? If we want crabs in this location, is it sufficient to protect that location? The answer is no. They may be coming from outer coast, Vancouver Island, up in Canada. We don't know where they're coming from, nor do we know where ours are seeding other habitats. There's an interconnectedness, transcontinental or trans-country boundaries and all kinds of things going on here of how these animals disperse, what they're doing, how they get there, how the animals behave. They can't control where they go laterally except by going up and down into different water masses. Sean Ariano in biology and down at Shannon Point spends enormous time out looking at larval behavior and then looking at water flow and modeling where these things are going just to try and figure out where these larvae might go, because it really does inform our conservation and restoration efforts. This is down in the Florida Keys. And you go down to Florida and you have no rocky intertidal, you have woody intertidal, which are the mangrove trees. There's a lot of, that's the only hard substratum for a lot of these invertebrates. And some work we are doing was looking at distribution of animals in these habitats and how they move from habitat to habitat. So this is an aerial view of the lower Florida Keys, Big Pine Key here on the right, and these little circles are mangrove islands. And they're the only places these animals can live. So the question we were trying to figure out is how do they get from place to place? When these animals spawn and these larvae come out, you know, that's 2-3 kilometers they have to cross. What's the behavior of the animals that allows them to do that? And these are some of the animals. These are called sea squirts again. They're great subjects for some of this work. And here you can see a bunch of them on a mangrove root, and they only occur on the roots. They're not anywhere else. And the way these work is they just suck water in one hole and blow it out the other, take the food out of it. But in the upper end of that, next to the arrow, is a brood chamber, and the little tadpole larvae are in there. And if you shine a bright light on them, one of them wakes up and swims out of the current. You grab it with a pipette and you can go out and release it somewhere and see what it does. And you put the pipette down inside there and get the very next one that's not swimming yet. And so you have controlled, some that swim, some that don't swim, that are the same size, everything else the same. And our question was, if you release those things, what are they doing to maximize their ability to get down to the next island? And we expected they're going to swim right up, stay in those surface currents as much as they can, keeps them away from the fish predators, and they'll manage to get down to the next island. And that's what they look like. The only reason you can do this is because they're fairly big based on larval size. So we'd release them. If they were not swimming, they'd kind of drift down, get down, and then a fish would get them. Release the next one and go down, and a fish would get them. And it was pretty dangerous for the non-swimming ones. So release the swimming ones, and they turned around and they attached right back on the root. They would not go anywhere. They're going, I'm not going anywhere. That's dangerous out there. They did not want to leave. And in fact, those larvae don't disperse at all. Everything they do is about staying put. So the adult releases it and it comes right back and settles right there because it's a safe place. That long distance is not a great way to go. Now, you might be going, well, who cares? You know, does that really change the world? Well, one day we're out working on our island looking at these things, and a whole bunch of divers came in, and they started snapping roots off these trees and shoving them in bags. And you go, what is going on? What's up? And we found out that a bunch of natural products chemists had taken these animals, ground them up, and put them on cancer cells, and found there's a very potent cancer drug in these things. It's a dihydroisoquinoline alkaloid. They named actinicidin 743. And because of the activity, they went to the National Cancer Institute and said, give us money to process this. We think we've got something. So the NCI said, okay, we want 1 gram of this product for you to do some preclinical trials with. A lot of things fall out in these preclinical trials because they're toxic. This did not. And so NCI said, okay, we need 5 more grams of this. So they contracted for these developers to get 6 grams of this actinicidin. 1 gram requires 1.1 tons of this animal. It was crazy. People were everywhere trying to find this stuff. They stripped it. They essentially removed it from the Caribbean. They're trying to get it out of Cuba just because they couldn't find it and they wanted it so badly. And nobody was going to synthesize it because it was so complex and it couldn't be monetized yet. And it became a race for people to try to get this animal. And the thought was, we'll take it off most of the islands and then they'll disperse, they'll reseed these islands, but it's not true. These things don't disperse. The absence of natural history knowledge here was really led to really bad management decisions. Could have been done very differently if we understood how this process all worked. Now, fortunately, eventually when it became clear it could be monetized, a group at Harvard synthesized it, and now it's marketed as Yondelis, very, very effective anti-cancer drug, soft tissue sarcomas and other things. So my point here is that knowledge of the life history could change a management strategy, which is not just about conserving the animal, but about the value of a product that could have come from this animal. And had they not been able to synthesize it, it may never have been able to come to market and be used. Okay, and then finally, the last piece of that, what about the juveniles? And all I'll tell you is we know less about the juveniles than we do about the larvae. By their very nature, they're yummy little small things that are easy to eat. So they're very cryptic, they're very small, so they're hard to find. We know very, very little about them. Deb Donovan over in the biology department with her students is working really heavily with the Puget Sound Restoration Fund, trying to restore abalone to our local waters. They have disappeared in the time I've been at Western. We used to see them routinely when we dived. You do not see them anymore. And they're trying to get them back. She's studying, what do they eat? How do they grow? How can we figure out what will make these things function? How can you outplant them so they'll survive? Jay Diamond down at Shannon Point is doing trying to develop some molecular tools that may also help us better address some of these problems. But juveniles is a black box. We know very, very little about it for any of these animals with these complex life cycles. So bottom line, thing I'd like you to go with, life history matters. Whether we're talking about Dungeness crabs and trying to manage populations. Or on the other hand, green crabs that are exploding in our local waters and we're really afraid are going to get out of control and cause some real problems. How do you manage and control those? Bamboo worms, this lower one, those are tormenting our local oyster beds and shellfish beds because they destabilize the sediments and the bivalves sink and they smother. And how do you get them out? How do you keep them? And part of the key is understanding those life cycles well enough that we can know what's happening and how they're dispersing and maybe how to control them. And various bivalves and many other animals we use commercially, how do we protect them? How do we maintain them? How do we restore them if they've been damaged? So my hope would be that after today, if somebody says to you, what's a crown of thorns starfish? You say, that is. It's very different than maybe what you see on aquanauts that my grandkids love to talk about. There's much more to it than maybe we think about typically of an adult animal that we're so familiar with. There's much more to the story and many, many interesting things to learn and a lot of important research to be done. So I will stop there and be happy to answer any questions you may have. Yes.
So why is it so hard to learn about the juvenile orchid?
Oh, so the question is, why is it so hard to learn about the juveniles?
Yeah.
The transition from the larva to the juvenile is, as you can imagine, it's a totally restructuring of the body. A lot of things change, and so it's hard to duplicate in the lab. We can get the larvae many times, but to get them through that step to metamorphose and become the juvenile is really difficult. And so we don't do it very well. So it's hard to do in the lab. And in the field, they're just so cryptic. You know, they're raining out somewhere, but we don't know where. They're landing somewhere, but it's got to be hidden. So we can't find them and we can't reproduce it very well. So the work with the abalone has been done in hatcheries, and there's enough motivation, enough money there. They've gotten through the process that they can't actually get them to settle. They know the cues. They can use enzymes of different types that cause the animals to do that, but most of them know. Yeah, please.
Do those crown of thorns have any natural predators, or is like, or is it just the coral is weak by human activities that are part of the problem? As like if these like historically the equator, would they be having this devastating effect? Do you think? You think that if that was the case, the entire supply of coral would be right now?
Yeah, that's a great question. Are there any natural predators of crown of thorns seastars? And if not, why did the corals not disappear based on the outbreaks of corals over time? First answer is they do have a natural predator. It's those big triton shells, large triton shells. And one of the arguments is that part of the reason they're outbreaking is because all the shell collectors have taken all the triton shells. I'm not convinced of that. There's better evidence that suggests that the outbreaks may have something to do with weather cycles, and you get more rainfall, you get more organics into the water, and that causes blooms of the plankton, and they feed better, so you get more of the larvae through that don't starve. As far as the question of why haven't they disappeared, the fossil record shows that these outbreaks have been going on for thousands of years, probably. You can go down through the strata and see large amounts of the crown of thorns. They haven't wiped them out probably just because they're periodic. You know, it's like, you know, so many examples of large carnivore predators that when your prey drops and your populations follow, and maybe that's all it is, or maybe they're just natural cycles where you don't have much food in the water and then they die back. Again, lots of questions we don't know, and wonderful opportunities to do really fun work. Yes, please.
Is anyone worried that getting rid of the crown urchins would like hurt the reefs in the long run?
Yeah, the question was, is anyone concerned that if they're out there shooting all the crown of thorns and killing them, it's going to affect the reef negatively? And it's a fair question. Anytime we get in there and mess with it, we tend to mess it up. We take out top carnivores out of habitats, and the deer go crazy, for example, and then you influence the bushes, or you take out the sea otters and suddenly the urchins go crazy and we've got no more kelp. I think people are so freaked out by the crown of thorns, they haven't thought about it. And corals are in such trouble anyway that it's sort of a rush to try to save the corals. So I haven't heard anybody weeping tears for the crown of thorns yet, but hopefully we're not going to regret this at some point. The other thing that's interesting, and this is more practical than anything else, is the funding for research like this tends to follow the outbreaks. And so an outbreak comes and suddenly there's lots of funding and then the outbreak dies away and no, we don't need to fund that. And so people get behind. And so to have consistent funding to really study it, it's hard to get because people respond to the moment. Please.
There's a question online sort of related to this, asking, have coral reefs evolved to deal with the crown of thorns population booms, or are they just unable because of their decline that's been happening?
Yeah, so can they respond to those booms? Have they evolved for that? I think they probably just tolerate it. You know, corals, their lifespans are so long and they're clonal animals. And so if you eat half a coral, there's still that genotype is there and they can grow back at some level. It's slow. So I think just for millennia, they probably just dealt with it and it would come, it would be an impact, they'd go, they'd grow back. Yeah, that's what I would think. I'm no expert in that, but that's what I would think. Please.
I understand that like a lot of invasive species funding is like a really inconsistent problem. I've heard in the past of other invasive species being put up on like the market as like a delicacy to eat. And so is there a way to use the market, or like are they edible?
Yeah, the question is, can you control them by marketing them? And the example you're probably talking about is an animal called the purple varnish clam. And if you go into our local beaches in the very high intertidal and you dig a hole in the sand, you can find very high densities of this invasive clam. And we don't know that it's really doing anything bad, but the Canadians renamed it the savory clam. So you go across the border and it's no longer the purple varnish clam, it's a savory clam. They don't taste very good from everything I've heard, but have you tried one? Here we go. So yeah, it's a strategy. These green crabs that we're so worried about, they might be edible, but they're really small and they're incredibly aggressive. Their species name actually means raving mad crab. They're not nice animals. And I don't know that you're ever going to market them because I mean, they're little, they're smaller. And so it's not worth the effort maybe, but.
How does the chemical formula in the vinegar affect the surrounding coral?
I think formalin's pretty clear. That's going to have some really negative things. Vinegar, you know, vinegar is not as persistent. It's going to break down more quickly. And so you certainly wouldn't want to squirt that onto the corals. But they're puncturing holes in the animal and squirting it in the body of the sea star. It's probably not great for the environment, but it's certainly not formalin. Yes.
For those of us doing invasive species ecology and economics, do you have any websites you recommend that data on different invasive species?
Experiments. There are a number of websites like Department of Ecology, Washington Department of Ecology has websites where it shows all the species of concerns, and there's a number of them in our local waters. As far as research being done with them, I think you'd have to get in the primary literature. So you'd choose the animal. If you look up green crab invasion and Google that in Google Scholar, you'll find a bunch of papers of the most recent work with that. But one of the concerns is that that species is moving south and it's right on the edge of Padilla Bay, which is the second largest eelgrass bed on this coast of the United States. And it's a critical habitat for fish, for the larval phases of fish, for many, many things. And so we know from the East Coast, those green crabs get in, they destroy eelgrass beds. They'll totally denude an eelgrass bed. And so there's a lot of concern about how do we keep these things back? How do we control them? What are we doing? But again, we're playing catch up. You know, I think 2 years ago, they caught 5 or 6 in the bay. I think this year it was dozens. And it's just going to continue to explode. Yeah, if you Google green crab, you'll find a lot about our local waters and concerns about it. Please.
Summer job, you be a green crab team, go around the state and catch a whole job.
Everybody hear that? If you want a job, green crab control. You show up, they give you a hammer. No, not really. They record data on them. They actually do research with them. As long as they have them, the scientists can use the data. Are you part of that?
Oh, no, my friends have done it.
Okay.
I just do audition.
All right. Did you have a question? Oh,
Yeah, so green crabs are invasive. Where are they sent from, like originally?
the green crabs are European green crabs.
They weren't brought here as like food.
No, and this gets to the whole life cycle thing also, that when a number of these come in things like seafood, it packs seafood and it brings things with it. And other places of ballast, you see the big ships move through. And when they come in, they're loaded down and they're low in the water, so they're stable. And then they unload their whatever they've got and it rides them really high. And it's not safe to go back across the ocean riding high, so they fill them with water from the local environment. Well, you get a whole bunch of larvae in these massive tanks, and then you go across the ocean and now you dump your ballast at the other end so you can load, and you've released all these larvae. Not only that, but the bottom of the boats get covered with all kinds of things too. So you're transporting a lot of stuff. Looks like we're out of time.
We're running out of time. We could probably maybe continue this offline in a moment. Brian, thank you. Next week, we've got John Harris, and the week after that, we'll be talking about Indigenous knowledge, and the week after that will be Deb Donovan talking about her work. So we will see you then. Again, students, make sure you signed in.